this session. And dear participants and my dear friends, a very good afternoon to and all. At the outset, let me thank the course coordinator, Dr. Rajiv, for giving me the chance to chair this session. I know that you are very enthusiastic about knowing about the knowledge on microscopy, especially on wide field microscopy. And in today's this session, we have the resource person or the expert, Dr. SANTHOSH Kumar, a scientist from Rajiv Gandhi Institute of Biotechnology, and he is delivering a talk on advanced applications in confocal microscopy. I hope you all will be enjoyed by his talk after the heavy lunch. Okay. Anyway, I hope he will narrate in a very beautiful manner. So I hope you will all enjoy. And I invite Dr. SANTHOSH Kumar for this session.
Thank you, ma'am. Before we move to the talk, a few words about today's speaker, Dr. SANTHOSH Kumar, who is currently working as a scientist in integrated cancer research at our Institute for Medical Science and Technology in the year 2000. He has received Young Research Award in 1999 from International Union of Material Science, also Innovative Young Biotechnologist Award in 2006 from DBT India, and he has been a task force member since 2012 at Cancer Biology DBT India. He is a governing body member at Amala Cancer Research Center, and he has successfully completed several research projects, and more than 25 research publications in peer-reviewed journals are also to his credits.
Basically, I will start with few remarks. In India, almost all of you know that bioimaging emerging as a big area, especially in Bangalore. But in this small city, even though we have a large number of institutions focusing on biological applications, research, Srijatra, RCC, and then obviously Biotechnology Department in Kerala University, still I assume that bioimaging is just is in infancy. Is not picking up, and in that effort, probably Srijat's effort to conduct such a workshop come seminar only on focusing the bioimaging aspects is basically a welcome approach. And again, the most importantly, it is always very difficult to find the right resource person for conducting such workshop in a very serious manner, because as I already explained, the kind of people available in India are limited actually. So we are very much fortunate to have Scott here actually. So with these remarks, I will start my talk. Again, one more thing actually. Initially, when he asked me to give a talk, I have suggested the topic of advanced confocal applications, probably assuming that I can talk on the Alfred application for biological applications. And later on, by looking at this program schedule, I realized that, in fact, Scott is covering almost all relevant topics, which biology people would like to actually. That's also, it seems to be, that means that he has done a great job actually. Again, this is only, you look at this video, since it is, as Madam has told, is a post-lunch section. It is going to be always a stress for everybody. So it is just to refresh your eye. What you are seeing here is actually, I don't have to, probably some people might have seen this video, isn't it? Okay. This is one of the wonderful biological events you are visualizing on live microscopy. Basically, the division of the cells, cancer cells, as and when it does divide, you can see that it's changing its color from red, then it is moving into yellow, then again it is reaching to green, then it is dividing into two. Again, after the division, the nucleus starts appearing red. Again, it is moving actually. So the red indicates that the cells are in the G1 phase. The yellow indicates that the cells are in the S phase, and the green, exactly the green, says that the cells are in the G2 phase. So this has been possible because of one of the two landmark discoveries probably. One is the discovery by three scientists, discovery of green fluorescent protein. I will show you a little bit of that. Even though I thought of talking more about the GFP, and you have wonderful talk by Scott on GFP as well. So I won't be going deep into that. Is it over?
No, not yet.
Okay. Now that is one of the wonderful discoveries. The second discovery is actually the Rogerson, his close associate Atsushi Miyawaki of Riken have showed that cells phosphorylate a lot of proteins while it moves from G1 to S phase and G2 phase actually, because of the differential ubiquitination of substrate proteins. So they have expressed these substrate proteins which are specifically targeted at S phase differentially, very precisely if I say, in G2 and S phase, and they express and put it in a cancer cells. So once it is stably expressed, as and when the cell in the G1, the green is degraded, and when it reaches to G2, the red is degraded. So in between there is a stage where you will find both green and red, that is S phase. So this is possible because of the two observations. One is obviously discovery of green fluorescent protein. Second is because of the knowledge that certain protein oscillate, and they identified the protein which oscillate and express in a systematic and scientific manner inside the cells. So with this actually I will move on to my presentations. This anyway I just showed basically because sometimes later on while talking about the specific area, I may be sometimes talking about this as well. This is basically, since you know confocal microscopy, I never will talk about anything, the technical aspects. This you know, but still to begin actually, this you know is a multicolor fluorescent microscopy image where different organelles are tagged with different colors, and you are you know observing under fluorescent microscope with unique light to the unique fluorescent probes which you are using, then collecting green, red and blue colors and overlapping in a wide field fluorescent microscope is seeing is wonderful. And then this is actually a two image of the same area. One is actually a wide field image, another is a confocal image. You know why confocal image looks good? Probably already you have been exposed to that. Basically in wide field fluorescent microscopy, when the sample is viewed under microscope, the white light or the filtered light is falling on your samples. Even if you have a very small thickness, it is completely bathing the whole thickness, whole volume of the samples. Obviously the emission is coming from the whole volume, and obviously the emission is detected by your eye or the camera from the whole volume. So there is a haziness, so there is an out of focus light is registered on your eye or the detector. Now in this case, in the confocal image, basically that is avoided simply because there is a pinhole actually. So that is making or completely reducing the out of focus light coming to the detector. That's why you will have a wonderful image. Obviously you remember this never will increase the resolution, but the contrast as well as to some extent resolution as well, but not very much. And that probably you will learn from the super resolution techniques. Probably Scott will talk to you later. And now the real advantage of this probably for the biologists is that one is obviously you have a means of cutting out of focus light. At the same time you have an equipment, confocal equipment, which is having an ability to move in the z directions, either objective or the stage, so that you can section the samples, you can collect the information from one plane, the whole thickness of the sample can be imaged. Obviously, usually single photon microscopy, we cannot go beyond a level of thickness, 100 microns, obviously possible. That is pretty good for most of the biological samples which we are using currently. Now beyond that, obviously multiphoton, you have a talk, multiphoton? Okay, it's there, you will become, okay, that anyway, it will be explained, it will be described to you later. Now, this also I can skip because I mostly focus on biological applications, and also to inspire how best you can adapt these kind of microscopy techniques for your application depending on your needs. And so that I will skip Okay, again, this is actually a fluorescent microschematic diagram is drawn over here. And in this case, as I told you, the thickness is depicted over here. When the light falls from the mercury arc lamp, this whole thickness is excited and the whole emission is going and the dichroic and reaching to the ocular. But in this case, what happens is that basically the whole thickness is excited here. But when the emission is coming out, and there is actually a pinhole, actually that is what determines the confocality. The pinhole can be open or closed. When the pinhole is completely open, this acts as a wide field fluorescent microscope. When it is closed, it will reduce out of focus light to reach to the detector. This is how confocality is maintained. And another major difference is that basically arc light is used here, but in confocal microscopy, this is very important, the laser is used, which is a point like source. And now this is very important. Point like light source is used means actually the image is formed not by a full excitation simultaneously, it is by scanning the area. For example, if you have a full image plane is like this is a scanner, the point like source, the laser, it is excite one point, and this moves, and then the point by point makes line, and then another line by line, and that create a frame actually. Again, this is very important for understanding some of the applications which I am going to talk. Then coming to basically, don't assume that confocal microscope has always comes with all advantages over the wide field fluorescent microscope. Wide fluorescent microscope is a wonderful equipment for a biologist because it have a lot of advantages which you never will find in the confocality. Even with the emerging technologies, I will tell you some of the things, some of the difficulties. One is that limited laser lines available. Nowadays numbers are increasing. Is okay. Sometimes white light laser is also has come into the market, whereby you can 478 to 670 any laser line can be tuned. These are changes actually, but still generally, but power of those lines will be limited. And acquisition speed, this is very important actually. Disadvantage of a confocal microscope, too slow to image many cellular processes such as calcium signaling events. Actually, so much of biological events which is happening in your biological samples. Fast will progress actually, so many things. So you wanted to capture those things means the machine which captures these images has to reach to beyond the speed of the biological events happening actually. Confocal microscopy, if you look actually, you have a machine called SP8, isn't it? SP8 from Leica, which is having a frame rate of 7 frames per second actually, 512 into 512. So with these frames per second, 1 second it can collect 7 frames. Some of the calcium signaling will go very, very fast. You need to collect one frame below 30 millisecond or 100 millisecond. So it is not possible with the conventional confocal. Again, a lot of changes are happening in the confocal microscope. Also, there are fast scanners like this resonance scanners. This is not available to your machines. And your machine, and then again, laser induced photo bleaching. Anyway, this is not very important because any light that falls on the samples, it is depending on the power of the light. It's obviously whether it is a white light or it is a laser, is going to have the same level of toxicity. And phototoxicity and photo bleaching are a serious problem. Now, see here, this I have shown because your workshop is also on the SP8, is in all workshops. So it's basically a spectral confocal microscopy. I have written something acousto-optical beam splitter, which is one of the unique technology available in the Leica. But please remember, this option is not available in your machine actually. But you have a lot of application package, and resonance scanner also is available with this machine, but it is not available in your machine actually. Okay. But spectral, this is one of the important thing, here, if you, let me see, okay. Here, okay. Spectral detector is available, and AOTF, which as I told you that In wide field fluorescent microscope, when the whole light is coming, when you put a specific filter, the whole light is falling on this everywhere. Wherever you focus, the light is falling on it. But in laser confocal, I told you that all the lasers are connected and integrated through a acousto-optical tunable filter, which acts as a filter which allow the selection of the line, as well as it allow the regulating the power of each and every line which is selected actually. So it is having a dual function, selection of wavelength as well as controlling the intensity. And while imaging, this is very important, while imaging, this is the only one very unique component which allow you to do a lot of biological application in confocal that is not possible with the wide field microscopy. And because of this, the laser, when it falls at a particular point, if you want to park a particular point high power laser, you can do. While simultaneously imaging, you want a low level of laser, but at some point or some application requires that you wanted to have very high power of laser to park at specific area. That is possible because this is actually fast responsive technology, and that is one of the uniqueness. And then this is AOBS actually shown over here. It is not available because you are using filter based system, but spectral detection is there actually. Spectral detection means it is actually all the emissions when the samples are excited with the multiple laser or single lasers, all the emissions are coming out actually. These emissions are collected and passed through a prism actually. The prism will separate the wavelength. Maybe the samples contains blue, green, red and all colors actually, but all colors are photons and it is not separated. And now you wanted to separate each and every color by separate detectors actually. Now the light is passed through a prism, the prism separate based on the wavelength, and it's a dispersion actually. You know, I don't have to explain much. When it is done actually, then there is a grid is shown over in front of each detector, and the grid opening and closing will allow you to decide what wavelength of light you requires actually. But remember, this kind of selection, basically most of the confocal microscopy and filter based microscopy, always there will be a filter wheel actually that allow you to selection the wavelength. So, but these two important things you remember, one is the wavelength selection is quite different from the wide field microscopy, and it is actually a point light source. AOTF will allow you to regulate the light as and when it image, and whether it can, this is very important for FRED as well as FRAP application, which you are going to implement on your samples. And for a typical example, this is actually an experiment of FRAP. You have a sample is actually labeled with multicolor. You are imaging as you have already started in workshop. So you are selecting all the required lasers, and it is exciting point by point, and it is generating. At this point of time, here you want to bleach only the green fluorescent protein. Assume that green fluorescent protein. That means that it will go like this, like this, like this, like this. That's the image, and it comes back. And as and when reaches here, you have already told the machine that I wanted to park the whole area to be bleached because of the high power laser. So as and when it reaches, AOTF will sense and AOTF will give and power it, and maximum light is delivered on this point. So it will bleach. When it reaches here, again it goes back to normal. This is actually, it's only, that machine will allow you to have a lot of flexibilities while doing bleaching and all those kind of things I am not telling. But remember, this will allow you to bleach while imaging, to bleach a specific area. This is very much important for certain application like FRAP as well as FRED. Now ROI intensity based imaging is also possible. FRED requires this unique property of a microscope, and photo activation is also another important area. Basically this also evolved from the discovery of green fluorescent protein actually. Green fluorescent protein is always green, but certain proteins were converted because of either because of the modification of the protein by mutagenesis, or because of the differential origin from, or a lot of other approaches were engineered actually to develop a new generation of green fluorescent protein that will show one color. At the same time, when activated with a particular laser, this will change its color to another. So this is also very important here. Why it is important is that if you large number of cells for biologists, if you for say especially I am working on cancer biology, if I want to track a cell over a period of 2-3 days in a very seemingly heterogeneous population, and you have a large number of cells expressing with photoactivable protein, you are activating a particular cell, I want these cells to be identified day after tomorrow, where it is going actually, what is its role. You are activating, and once it is activated, it changes the color actually. Now next day you image it, you find where is it, where it migrates to brain. As our example, if a multiphoton is there, where it migrates to brain, it metastasis, or what is its functional role to interact with other cells, all these kind of things, telophocytes, infiltration. All actually, if there is this, I am telling, if there is a technology, is remember the technological potential, and then you decide in what way this is going to help your application, and just use it. This is one of the use I said, but this can be used for diverse applications. Only thing is that as and when it activates, it changes its color. And here also, photoactivation actually needs the laser, a large number of cells you are imaging, but you wanted to activate with a particular wavelength of laser, only decide predefined cells. So you need, that is what. And then for all kind of applications, when it comes to biology, real long time imaging, you again require certain, because some of the application I am going to talk to you basically are live cell compatible. So you need to have a system to maintain the cells on microscope on live for a long time. The long time means how long time, and probably 24 to 48 hours is the decent time for certain events tracking. Calcium imaging don't have to do it, it will end with the 5 minutes, but it's aggressively fast. But slow moving events, like for example, you saw cell cycle in the initial video that has been captured over a period of 3 days, 3 days. So you need to maintain the cells on microscope for 3 days. So there must be, obviously almost all the mammalian cells requires specific temperature 37, carbon dioxide 5% to maintain the pH, and obviously humidity also needs to be maintained. So the machine always will come with a air device. Different kind of devices are there, either your microscope, you have an actually on-stage incubator, isn't it, from Tokai Hit?
I think all they have is
Huh?
I think all they have is a heater here. I think.
Only heater. Oh yeah, no. Later on they purchased.
I think they have CO2.
Tokai Hit, huh? Not there. Okay. Yeah, I think separately there was a.
There is a separate unit is there, but not started.
Oh, you can put it anyway, anytime.
Purchase carbon dioxide.
Okay. Anyway, this is one of the system. Basically, the whole microscope is incubated in an incubator actually. So the whole inside is a bubble with a free mix air, which contains 5% carbon dioxide, and also temperature. The whole area is heated, and so that this one, and actually this is very difficult to maintain actually. When you might have seen it microscope, especially confocal, you put hands to have it's little bit difficult. I don't like this, but we do have a machine like this. And one of the best incubator is actually this is a biotech. It's very less expensive unit. You can create. It's a very small unit. As and when there is a requirement, you can put using our slip. You can create an environment. It's very less costly. But there are actually other incubators like this. Actually, this is actually on-stage incubator, predefined. This is the one probably you might have processed from Tokai Hit. And this, the cells are on the chamber. You don't have to heat all the. This is a logic actually. You don't have to heat the whole microscope. Only you need to maintain the temperature for the cells. So the 37 dish are kept on the incubator, which is placed on the stage, and it is heated, and as well as there are regulators of to bubble the pre-mixed gas, and humidity regulation also. But one of the problem of this unit is that compared to the other one which incubates the whole whole microscope, there is a temperature drift actually because especially when high magnification you are imaging, even if it is placed in the incubator, the objective is actually hitting and touching the sample while focusing, and then the heat, it is heated, and the outside microscope is not heated actually. So there is a significant drop in the temperature through the objective, through it diffuses towards the body, to the body. Obviously, the microscope you will be keeping in the AC. So this is actually long time stability of temperature maintenance is a problem for this one. And again, this is very convenient. Anyway, this is depending on this. This is also another one similar, but here you can put 96 well as well. Okay. Now, another major problem, whether I don't know whether your machine is having that that unit. Another serious problem in live imaging for a long time, as I have seen on the image previously, is that even if you focus, you want this focal plane to be maintained for 3 days. It is extremely difficult for any machine actually, because there is a thermal drift and there is little bit change, very fine change in the z direction is damaging, because the cells will go out of focus actually. So there must be a mechanism to keep the focus intact actually over a period of time. There are a lot of technologies, adaptive focus compensation from Leica, perfect focus compensation from Olympus, ZDC from Olympus, PF, this all will work on a same principle actually. They use actually a 633 or 750 770 laser, and this laser will hit and it will memorize that depending on how far it moves actually. There is a dichroic, specific dichroic as well. This will memorize that actually, then it will autofocus, maintain throughout the experiment. Okay, that's a little bit, even though it's very simple, it is one of the expensive units for a microscope. Again, this I don't have to talk actually, you have seen it. With this few introduction now, why real time? Real time means when a wound is made, you put under microscope after 24 or 48 hours whether the wound is healed or not healed. I have some videos, anyway it's okay, it's not very important. You can, after 24 hours you will find that is completely healed actually. So the dynamics you can very well see. This is the advantage. Then coming to a biologist, advantage of microscopy. When microscopy is actually, when somebody is beginning to understand, they obviously will find this as an equipment to resolve fine details visible to your eyes. So you want to see different things. Obviously, immunostaining is the first one. You stain the samples with the protein of your interest and using a secondary antibody, stain with the different colors, and you view under the microscope. You will find like this. Basically, this is a conventional microscopy application. And in this, if you talk about this, you can elaborate in any manner actually. Any kind of proteins can be visualized, especially provided you have an antibody and you have a secondary antibody conjugated to a fluorescent proteins. Maximum of 4 or 5 colors, it is extremely easy to image use in wide field microscopy. 4 colors is not difficult, 5 color probably a little bit difficult because of the spectral overlap and all this, but people can do it actually. Now confocal also will do the same. Then this is a conventional imaging, and then coming to live cell imaging. Actually, when you look back molecular probes catalog or Biolegend catalog, you will find large number of fluorophores or fluorescent dyes, which they claims to be specific for unique certain applications. For example, there are probes for calcium. There are probes for pH, there are probes for so many things now. Probably any biological molecules, if you wanted to look on this on your samples, sometimes you might see fluorochromes specifically for that in molecular probes. Specifically, they say that it is very specific for it, and different color images. So obviously, you can use it. At the same time, if you look at the literature, every year large number of fluorochromes or fluorescent proteins or nanoprobes with the fluorescent properties are developed and reported to be essentials for diverse applications. This is an entirely different area. You can depend on that, read it, What is the biological readout you want? You read it, look for, there is a dye, there is a probe, and then look for your machine whether the probe, what is actually the output it will show, what kind of color it will show, and whether the color can be detected with the system you have here, use it, fine, there is no issue, until unless you specifically substantiate that it is very specific for the molecule for which it is intended, fine. And for that you have a large number of I won't talk about that because it takes a lot of time. I cannot, because anyway probably you have an extensive sections, somebody will talk on those aspects. And again, if you look, these are, you must remember, for nucleic acids, stain also, you have a large number of dyes, credin orange, chacran, diazole orange, or ethylene bromide, with the different colors, which are excited with the different lasers. Look whether this excitation light laser is available with the machine, and also corresponding filters are available. Then obviously you can use it. DNA probes. This is the pH sensitive probes, oxidation sensitive probes. Then, so this is probably you people will be well aware of this. Actually, most of the biological PhD students in biology, plant or zoology definitely will use this dye. This is the dye people used to claim that it is actually a sensitive for ROS. A lot of problems people might have also faced while using these dyes. Anyways, okay, I won't talk about that. It's fine. If you are getting good results, it's great. But this is not considered to be a good probe for ROS detection. But still, publication, if you look, you will find, I think this is also developed by Rogerchen, probably. Then specific organelle probes also, large number of cellular organelles, cell biology, plant cell biologists or animal cell biologists always would look for the organelles inside the cells. Either you can stain with the after fixation, or you wanted to see the live cells, you have a live cell specific dyes with the differing spectral colors. So you can use any of those, it's fine. And this aspect actually is very important. Calcium cannot be visualized directly, but calcium is a very important second messenger, which is known to play a lot of functional roles in plants as well as cell biology and zoology, as well as disease biology, heart, neuronal memory. So it was always fascinating for biologists to find better probes to visualize calcium. So I will explain some of the examples. That is why I have shown over here. I told you that there are calcium probes, Fura, which is also developed by Rogerchen. But a lot of Fura is a very nice dye, provided you have a system. It is actually a dual excitation dye actually, okay. You have a system to demonstrate it is fine, it will work very well, provided you load it very well and your cells will respond to the loading very well, otherwise it's a problem. So these problems anyway. Calcium as such doesn't have any fluorescent property, or is very small, and also obviously as such in a microscopy or anything, if you wanted to see as and when happens, there must be either a visual readout, fluorescent color, or color as visual, or some other photon emitting, which so that you can use for X-ray, something like that is there is fine. But as such, they doesn't have a fluorescence or any color emission property. So that is why as such you cannot see. Yeah, fluorescent probes are there. As I said, actually, what I meant is that as such you cannot see actually, because cells, for example, cells you can see because they are little bit bigger, as well as with the kind of contrast technique you use, it will show its contrast, phase contrast or DIC. But this calcium is a very small molecule which goes below the size of 1 nanometer. Is extremely important, difficult even to see with any microscopy technique as such. But provided it is having a indication of color to see and quantify it is obviously possible. But that's what I said. For that there are dyes actually. The dyes what they will show Fura, it will show is that Fura will bind to high calcium and low calcium, and show an emission in the green fluorescent color actually, but the emission, the excitation spectrum will change from 340 to 380, okay, depending on the concentration. So, you can see that when you excite with the 2 color and the emission you collect over a period of time, you will find that the excitation property will be changing actually, but I do not have an image here to show. So, as and when it happens, you can see the ratio change actually. It is very good dye. People have a lot of dyes are there. Similarly, this is excitation, and Indo is actually a dual emission dye that is also widely used. Now, and again, these dyes were actually, most of the dyes were UV excitable. So, that is also damaging to biologists. UV excitation is not very good for biological samples. Now, this is fine. With this actually I will try to let's give an overview of green fluorescent protein, what is it, and actually this is a protein isolated from marine organism, jellyfish, Accoria victoria. Okay, this is organism. This as early as very olden days people know that the jellyfish when washed ashore, people used to, Japanese people used to collect this and they used to put on the walking stick actually. In night actually, when the electricity was not there, they used to show this on dark so that it will glow, fluoresce actually, green light is coming out. So, they were using as a torch actually. So, some of the scientists, especially I just will show you. Osamu Shimomura was so fascinated with this unique color, light producing protein organisms, and later he has purified a particular protein from the marine organisms, and he called this as a green fluorescent protein actually. And basically then he demonstrated that this as a protein probably in 80s. Two other scientists, Douglas Prasher and Martin Charfy and Roger Shane, happened to hear about this discovery, the discovery of a protein from a natural source which fluoresces. So, they were actually studying at those time to the gene expression on diverse organisms, including mammalian cells, plant cells, Rhabditis elegans. They were trying to see, as you were just mentioning, whether we can see something. They are trying to see protein expression on the cells, but it was not possible. Every time you have to kill the animal and stain it actually. Live cells, it is not possible because you cannot see any protein actually as such. So, since they happened to heard about the technology of green fluorescent protein, he said that it is a protein. Then that time it was actually genetic engineering approaches were just evolving. You can clone any gene of your interest and express actually. So, then these 3 scientists realized that they thought that, so if it is a protein, there must be a gene. If there is a gene, you can generate the gene and you can express in any cell. So, that is what created the revolution of green fluorescent protein actually. So, Roger Chen has cloned this gene from the marine organisms and expressed in diverse cells. This is a bacteria plate. And what is the beauty of this is that this technology is that subsequently the same group active now still Roger Chen is actively involved in developing large number of fluorescent proteins. Probably each year you will find large number of discovery of different color fluorescent proteins from different organisms nowadays. Nowadays a lot of people are active. Still Roger Chen is one of the leader in this field. And they subsequently created mutagenesis. Mutagenesis also is one of the, I don't have to talk about detail here. As and when you change the amino acid sequence, they realize that its color properties or spectral properties will vary. And depending on the knowledge, they develop large number of color variants. Now, you have cyan, blue fluorescent protein, cyan fluorescent protein, green fluorescent protein, yellow fluorescent protein, teal fluorescent protein. Number is too much increasing. Is unimaginably very high. And what is actually the advantage of this technology to a biologist? This is what we will learn from next slide. And obviously you can, as I told you, now it can be expressed in any cells, we will see actually. And again you can express with any gene of your interest, because you know your gene, you know your nucleotide sequence, you know the nucleoside sequence of green fluorescent protein, and you express the gene in such a way that your gene comes first and then green fluorescent comes first. Otherwise, second. Otherwise, you first clone GFP and then your gene of interest. So, the gene when you express inside the cells, it is expressing, it is protein is synthesized. As and when the protein is synthesized, the most remarkable thing and fortunate things happened is that most often this behaves as that of the original protein. That is the beauty of this. If it would have been not, most often it never will happen, but it so happened actually green fluorescent proteins. And so that you are actually tracking your gene or protein of interest in live cells. So, as and when the protein is expressed, then you will, depending on the protein where it goes, there you will find fluorescence actually. Now, with this introduction, again, it can be subsequently, it has been expressed in large number of organisms. Is see this mice, the green fluorescent mice. Even in India, we have a green fluorescent mice developed by Panikar in IAC, and then this is actually is not very important. So you can see it actually. As and when an animal is developed, if it is a constitutive promoter, it will be expressed in all the cells in the body. So embryologic studies, it is a great advantage actually. You don't have to kill actually. If a multiphoton microscope is there, you can see all the cells actually, how it is organized. Even in a tissue wanted to grow in the lab as well, you can see it. That is the beauty of this. And with this, and again keep this in mind, any number, you have a large number of fluorescent proteins currently available with the different spectral properties. The spectrum is not very important. And because of this great discovery, they shared the Nobel Prize for chemistry in 2008. This is not very important. This you will find that now. This is also another protein which is basically not green fluorescent protein. It's IR emitting proteins which is expressed in the frogs. Now there is a company in Russia, Rogen actually. They are more specialized in developing IR specific dyes. This fluorescent protein, because green fluorescent protein obviously you can see it, but a lot of problems are there when you use as such in animals for imaging, because tissue absorb a significant amount of white light actually in the visible range. So that is why IR specific fluorescent proteins are also developed for certain by animal imaging applications. Okay. What are the conventional applications of this? Yeah, for gene transcription, if you wanted to temporally study the transcription of a particular gene inside the cell, at which point it will transfer. You express the particular protein with respect to either put with the GFP or whatever green fluor or other variants, and then look under the microscope what is its behavior, whether it is going to the nucleus or some. This is actually a conventional experiments. Any proteins can be tracked over a period of time for its behavior. This is any protein actually, whether it is the plants or whether it is an animal or whether it is in another organism, it is possible. And certain things, this is fine, okay. Now, so this I don't have to explain more, because you will be learning more and more about that in the course. And for a biologist, at the same time, most of the signalings are contributed by not only by the expression but by the protein-protein interactions. Protein-protein interactions, since you all are, I assume that PhD students probably, you always will wanted to look for protein-protein interaction studies, and lot of techniques has evolved, label-free SPR technology, protein B, immunoprecipitation, all this is fine actually. But none of this technique will allow you to study a protein-protein interaction in subspatial level. In a cell, exactly where you want to know, in a cell where exactly my protein of interest is interacting with another protein actually, okay. This is impossible to do with any of the protein-protein interaction biochemical assays currently available. Now, microscopy, is it allow you to do that? No. The reason is that any protein can, as I said, any protein can be expressed with green and yellow. Okay, green and yellow or green and red actually. So, two proteins of interest if you wanted to see whether it is interacting. Obviously, you assume when it interacts, it has to come closer. Okay, whether this will tell you that it is interacting? No. The simple reason is that why it is. Being microscopy students, probably you might have learned it. Why it is? Because no, not that actually. Two proteins. I have a protein, B protein. A is black, green, B is red actually. A and B when comes, it will become yellow. Is it yellow that indicates its interaction? No. The reason is that the size actually. The size of a protein is always 5 nanometer or lesser than that if it is in the tertiary structure. For two proteins, when it comes and interact independently as two molecules, you need to have a device or a system to resolve 5 nanometer range, 5 or 10 nanometer range. But none of the microscopic techniques will allow you except some of the super resolution technique, which will be explained to you later. And now, so that is the reason actually our best microscopy will have a resolution limit of around 220 or 240 nanometer. Never will allow it to. Now, in this case actually a protein-protein interaction study by microscopy was developed based on the physical process proposed by Theodore Foster in as early as 1914. And basically, if you know, the real interesting thing is that this is a typical example. Two proteins is somewhere you will find colocalization, but these two proteins are not supposed to interact. This is a BCL2, this is a mitochondrial outer membrane protein, but you will find that somewhere there is a close association. It is not supposed to interact actually, but still you will find that it is colocalized. That means that it is not resolving that level. And now this is actually the reason. FRED actually is a physical process whereby two fluorophores, when it's a fluorescent resonance energy transfer actually. While studying with a large number of fluorescent dyes, Theodore Foster realized that when two fluorescent dyes are coming very closer actually, when you are studying the fluorescence from different combinations of dyes, you realize that certain dyes, when you mix it actually, one dye will emit one color, another dye will emit one green color, another kind of red color actually. Basically, when you mix it, when you excite with the red, green, he was not getting the green, rather than he was getting more of a red actually. So he realized that basically something is transferring the energy to the next one. So while with the very precise experiment, he showed that this will happen when fluorescent dyes or anything when comes very closer, as close as to 5 nanometer or 10 nanometer range. The first one, instead of emitting its color, transfer the energy to the second. So that instead of getting the emission from the first one, you will get the emission from the second one actually. So for example, if you assume green and red. Green when you excite green, you are supposed to get green color, but instead of getting green, when it comes closer, you will get red actually. And what is importance of this is that this will happen only when they will come as closer to 5 to 10 nanometer. Lesser, even it comes beyond 1 nanometer and some other energy transfer will happen. But when it goes beyond 20 nanometer, no transfer will happen. So based on this knowledge, physical process, microscopy has evolved this technology, even by Roger Chen as well as lot of other scientists, to extend this knowledge to study protein-protein interaction in biological samples in subspatial level. And again, while doing so, this will allow us to generate large number of probes to visualize large number of signaling in a precise manner, including calcium. So I am going to talk about that in the coming slides. Remember, this thing, okay. A typical example for what are the applications now these were used. Monitoring interaction between this, see these when you read these, monitoring interaction between macromolecules, protein subunits, DNA oligomers, conformational changes of protein, association and dissociation of protein and subunits, changes in the concentrated small metabolites like CAIMB ions, and also energy in ATP and membrane potential change. All these can be very well studied in live cells using technology. Now, how this is done is, for example, if you wanted to study a conformational change of a protein using this, you express the protein, a single protein, one of the terminal you use a donor protein, another C terminal, N terminal use another protein which is an acceptor actually. So, keep this in mind, the FRET will happen only if the emission from the first donor have a significant overlap of the excitation spectrum of the second one. So, the emission from the first has to have a significant overlap to the excitation of the second one, then only it will happen. So, here your task is to select the right donor and acceptor. So, keeping this in mind, take a typical donor and acceptor. When it is linear, what happens is that when you excite the green, you will get green emission. But when there is a conformational change will happen, if it bends the one terminal to other terminal, because its bends means the molecule having a size of 10 proteins have a size of 10 nanometer. When it bends, most often, not always, most often the two green and yellow, the donor and acceptor come closer. So that what happens is that it will transfer energy to the acceptor, so you will get emission from the acceptor. Is it clear? If at any point of time, if it is a little bit, I am going probably fast. If you have a doubt, please ask. Okay, anyway, this is the intention is to give an overview of the potential of this technology. So, you even if you are not getting the exact scientific concept behind this is also fine, but these are possible actually. And again, protein hydrolysis actually, this I will show you some of the examples. And then ligand receptor interaction. Yeah, receptors actually, most of the receptors, ER alpha, ER beta, hormone receptors, growth factor receptors, all in biology will show its function when a ligand will come and interact. And such an interaction can be very well studied by this technique using microscopy. Now, how this is done, I just will show with an example. See, Scott, you will be covering that acceptor bleaching, all this,
We talked a little bit about it today.
Little bit, yeah.
So they got an idea a little bit.
Okay. So, one of the widely used, how you will show actually, you have a microscope here which is having a unique application we saw to show FRET. It's called acceptor bleaching. Acceptor bleaching works in a very, very, very simple way. Like for example, you have assumed that you have two proteins. One is expressed with the, assume here it is the blue color. Another is you have expressed with the yellow color. Okay. Now, assume that if they are interacting, means it is coming closer to 5 to 10 nanometer, they must interact actually. When you excite the blue, for the excitation for the blue, 452, it actually will transfer the energy to the yellow and you will get emission from the acceptor. But at the same time, you will see a decrease, since it is transferring the energy to yellow, you will find a decrease in the blue. Now, in this ideal situation, you are imaging it actually, you are exciting blue and you are getting a yellow color. That means it indicates that it is actually transferring the energy. But obviously, you must remember that a lot of weakness of the microscopy system actually is spectral overlap. All these also will excite this one also. You don't know, the cross excitation is also generated. So, that has to be corrected. How this will corrected in the Leica machine is a perfect technology called Acceptor bleaching. Now, this is fine, it's under 10 nanometer range it is coming. After taking an image of blue and green, what you are doing is, this is what I said, you are exciting, transfers energy, you are getting yellow, then afterwards you are in the same space, you are identifying an area where you are asking the machine to bleach the only the yellow color, then leave the acceptor. This is possible because what I said initially, AOTF will allow you to park the laser light high power in anywhere, specific area. So you are bleaching it. What happens when you bleach it? When you bleach immediately, what happens that previously there was some energy was transferred to the acceptor. Now the acceptor is deactivated. Now there is no one or nobody to accept this. So whatever it was transferred to previously here will move back to that. So you will get increase in the blue color. This is one of the very sensitive technique to show protein-protein interaction. A typical example. Anyway, this practically it will be shown to you. See this two protein in a cerebral tissue. This is actually an image of donor, one protein. This is an acceptor protein, and in the same area you have taken an image of both donor and acceptor. After that, immediately you told this machine to bleach this area, only this area. After bleaching, you are taking another the same imaging conditions. So after bleaching, what happens here? Only this area, the same imaging conditions were maintained, but only wherever you bleach, you have an increase in the green. That means that this much was previously transferred to the acceptor because of the interaction. Now it is lost. So based on this, you can, what is the beauty of this is that you can quantify the interaction potential, and also you will find the heterogeneity, spatial information of the interaction. No other technique will allow you to do that. Okay. Now another issue. Okay, this is not very important. Another application I am just talking about you now. Oh yeah. No, I didn't get actually. No, you mean subspatial differences actually? Yes. No, assuming that here in this case actually, for example, when I bleach here, if there are problems. There are some problems I will tell you. One of the problem which you have is that if a particular protein is spatially not abundant in some space, then it is a problem. But if it is uniformly distributed, this is not a problem, because the protein is a soluble protein inside the cell, obviously will be uniformly distributed. It is unlikely to compartmentalize. Now A and B is uniform, assuming that there is an ideal situation. That time you will get a pattern of increase in the fluorescence in everywhere. Then ideally you assume that it is everywhere it is interacting. Sometimes even with both the proteins are soluble, but you are not getting an increase in everywhere uniformly. That clearly tell you that the interaction is spatially heterogeneous. Is it clear? Okay, that is clear. So that is, there is no doubt. Now the second assumption, if one of the protein is soluble, another protein is targeted actually. Okay, it's not soluble and it is not uniform. So when it is in some space it will interact, organelle it will interact. Then also this will allow you to tell actually whether some organelle is interacting, in the other space it is not interacting, because this is actually the ratio you will get actually is like this actually, the Fret efficiency will, this there is not much. It could be two reasons. One is actually the microscopy resolution also will contribute for this. And second issue is that the other protein is not absent, is a problem. When it's staining, when it comes, the second protein is not uniformly stained, also problem will come. Problems will come, but these problems are very less. Is it clear? Huh? Now another typical example of this application, I will tell you that citing the example, in enzyme activation, a lot of enzymes were activated inside the cells when cell leaves and when proteases cell proliferate, and also when cell sometimes dies, proteases are activated. Proteases, the unique characteristic of proteases is that proteases, when they are activated, they will cleave proteins after specific amino acid sequence. Casein will cleave a particular protein after particular sequence. Similar to that, caspase, which is an enzyme which is activated during cell death, that also will cleave after specific amino acid sequence called DVD actually. Now, this technology has been adapted to visualize caspase activation, diverse number of caspase activation inside the cells. Example is a caspase activation, caspase 3 activation that happens during apoptosis. Now, what has been done is that you express a CFP cyan fluorescent protein and yellow fluorescent protein. In between, you place a activated caspase specific sequence, which is cleavage specific sequence. So, when you express inside the cells, when you excite the cyan, it will transfer energy to yellow because it is actually a linear protein. Now, in this situation, when the caspases are activated, what happens? They will cleave the sequence here. Okay. When it cleaves, the two proteins become spatially separate. So, now I will show you an example. How exactly? Is this clear now? This is actually a cell which is expressed with the FRET probe CFP and OFP, but actually what I am showing here is that both the color are merged actually, cyan and green were merged actually. So, after some time, 7 hours, 8 hours, what I have done is that I excited with CFP, then collect CFP and OFP. Then we added a compound which will cleave activate caspase activation. So, after 8 hours, one cell has changed its color to blue. It is a real color, it is not ratio image. Ratio image will be much more dramatic. It is a real color because in these cells caspase got activated because it is a cleaved. So, the did not transfer to green is happening, so the blue has increased. Now, as and when it goes, more and more cells are becoming blue in color. See that? And enzyme activation inside the cells in real time you are monitoring. I have some videos because I do not want to lose time. So, okay, we have time, some more time. I will show the videos later because one by one I will play it later. Is it clear now? So, any kind of enzyme, for example, you have a calpain enzyme, you have a casein, you have another enzyme trypsin. Any enzyme activity you wanted to visualize in real time. If you know the specific sequence, then you can use this technology. Use one acceptor, another donor, in between the specific sequence you put it, express inside. What is actually the advantage of this technology is that for screening application is that the enzyme activation most often is very promising for therapeutic purpose, activating enzyme or inhibiting enzyme. So, for drug screening it has been very potentially used. So, one of our area is actually to, as I told you, mentioned here in the initial introduction, we have been working on developing assays for high throughput drug screening. So, now this is not very important, acceptor bleaching, all this is not very important. Again, similar to that, this is also Rogersen in 2002. Basically, remember all these technologies were actually developed by Rogersen. These approaches were actually. He described one of the very powerful technique to visualize growth factor activation. Growth factor activation, why it is important is that when a cell, when you add a growth factor, when it goes and binds to the receptors, immediately tyrosine kinase is getting activated. It's a very fast event actually. So, people were always would love to know the real times to see the growth factor activation inside the cells actually. So, he expressed a substrate peptide as well as phosphor tyrosine binding protein with the CFP and OFP, and then when ATP or phosphatase is added, when this is activated, they undergo a conformational change. When the kinase binds to the in ATP binds to this, and this will undergo conformational change. This is actually a conformational change sensitive protein, the sensor, but it reflects that activation of a growth factor. When it undergo conformation change, since it is linear, from linear to it will bend and come closer. So, FRET will happen as you shown over here. See the beauty of this. There is no growth factor. After adding the growth factor, the FRET is happening in compartment, cell membrane, compartmentalization, progressing very fast, then moving, see that plus. In CC the time 0 minutes, 15 minutes, 21 minutes, 36 minutes. A growth factor signaling transduced through the cell inside the cell is first time visualized in real time. So, these are the real applications of this. Now, coming to the calcium signal, just like that, a significant quantum of amount has been invested by scientists to develop real time tool for seeing calcium imaging, because I told you that this is one of the very fast progressing events once it is initialized. A lot of problems people have already faced with the dye specific for the calcium. So, Atsushi Miyawaki and also Rogersen realized that actually they express two proteins, calmodulin as well as calmodulin binding domain of M13, and they express these two proteins with the cyan and yellow fluorescent proteins. When calcium will binds, they know that using in biochemical studies, they know that calcium binds to this, they will undergo conformational change and bend actually. So, using this technique only, they express a linear protein of calmodulin and also is binding component M13 as a linear protein, and in the linear protein they express one end cyan, another end yellow. Then what is actually the beauty of this? When you express this on say cells, after a signaling is happening, the ratio is, it's a very fast progressing event, ratio is significantly increasing. So, this is called a chameleon. It's called a calcium chameleon, because chameleon is an organism which fast changes its color in response to any environment, external aggression actually. So, that is why they call chameleon is one of the wonderful probe for calcium imaging. And another beauty of this is that since this is actually a fluorescent protein, it can be targeted to any organelle. It can be targeted to nucleus, it can be targeted to my cell membrane, it can be targeted to mitochondria, it can be targeted to ER. While doing so, they act as the sensors of calcium at this particular organelle. And with this actually, this is another image is one of our images. See that this is also a real image actually, the ratio image. You see the mitochondria, we have expressed mitochondrial chameleon in the mitochondria. Now certain cells ratio has significantly increased. It's very far. I have a video, and now I just will show to some of the videos. And this is okay. Anyway, I will show the video. So you can target it to any organelle, cytoplasm, so that it acts as a cytoplasmic sensor of calcium. It can be in the mitochondria, it can be in the endoplasmic reticulum. Then actually, this high throughput adaptation anyway is not very important, because this is some of the technological achievements not very important for biologists. And again, I told you that this initial technology I told you now, which was developed by Atsushi Miyawaki, allow you to study the progression of cell cycle, because the nucleus will change its color from red, green and oscillate. So, that will allow you to tell you the cell cycle status. In biologists, for any biologist, it's two important phenomenon which is always more very interesting, is that one is cell cycle, another is cell death. So, we try to couple these two things. I told you we have a probe for caspase activation using FRED, and we also have a probe for cell cycle on the nucleus. Fortunately, it is on the nucleus. So, we just try to couple both. Now you see, I don't know, see this, basically the cytoplasm, the caspase sensor probe is there. The nucleus, when you look, tell you that cell cycle stage. So, now using these approaches, you can visualize in live cells cell cycle progression as well as cell death. I just will show some videos with those. I can probably this is, you see this after some time, some of the cells are actually changing color to blue. It's a real color, it's not ratio. Basically is becoming blue because caspase activated the FRET test lost, so it is blue is increasing, and after some time more and more cells are becoming blue. Now some cells are still surviving. What are the implications of this for us cancer biologists? That at any point of time when a cancer cell is treated with the drugs, we wanted to see how many cells dies, how many cells will progress, how many cells persist actually, and what happens to the persisting cell. These were the questions always. That is why we developed this tool. So you see that at the end of that, certain cells are still not dying, no caspase activation is happening. Again, we have developed several other probes using these techniques. Actually, I won't, because as I told you, the number of fluorescent proteins are increasing tremendously. Anything can be used as a donor and acceptor. We are changing because of depending on the other parameters we are looking. So again, what is actually here the beauty of the confocal microscopy is that, see, this is actually how many this is actually a tumor mass which is grown in the lab, which is showing that CFP and OFP of Fred. But you cannot, even in the 3D culture, you cannot quantify how many cells are there, because cells are very closely packed actually. So what we have done is that we express with an NLS sequence, the same probe with NLS sequence actually. When it is done, you will find all the cells can be quantified in a confocal. So the whole section, you will find that how many cells are in the even in the tumor, how many cells are dying, how many cells are surviving, how many cells are in the G1 phase. All this can be very easily quantified using the images. See, this is a ratio image. The ratio has significantly changed in that, but there are some cells which the ratio has not changed in the 3D tumor after treatment with an anti-cancer drugs. So this survival, all this, then again you see here this image. What is shown over here is that a growing tumor. There is nothing is seen in the middle. This is actually a unique characteristic of a tumor which grows even in the body. When tumor grows ascendance bigger, it cannot find the oxygen and nutrition in the deep, so they start dying actually. When they die by necrosis, nothing is there, it's completely disintegrated. So those are, this is the animal, even animals you can express the tumor development and metastasis using these models. This is actually an initial tumor. After some point of time, you see the tumor has spread throughout the body of the animal actually. So these are the clear applications of with this actually anyway I won't now, the Fred and Frapp anyway I thought of talking, but it's okay, not very important. With this I will stop now. I will show some of the videos probably, and okay, just you can ask questions. I will show you. Clearly look at this is caspase activation, the calcium in the mitochondria. See this, remember this we have added a calcium inducing stimuli, and you see how fast 7, 8, 10, 12, 13, 14, 18 minutes. 18 minutes we image actually, and it's very when it starts is once it starts it's fast progressing. This is what we wanted to capture. Confocal microscope never will allow you to do that. Conventional, because you need because the simple reason is that 100 frames, this is actually 100 milliseconds. Every frames were taken in every 100 milliseconds. So to 600, around 600 sequences over a period of 12 minutes. Confocal microscope, usual confocal microscope never will allow you to do that unless you have an ROI scan. Or ways that the better method is to have a resonance scanner which goes fast. And seeing is believing. Some of the cells, if it is not calcium is not activated, also you can visualize. This is a cell I told you that this is allowing to, this is, the nucleus you look is changing its color to green, red. What cytoplasm, what is expressed is that Fred probe for caspase activation. It is not treated with any drug, it's a control. They are growing very happily. Now cell is dying, there is no bluish color at all actually. The same cells when treated with a drug, I just will show you. So the message is very clear actually. So how this is image is that we are exciting CFP, we are collecting CFP and OFP, separately collecting the red nucleus, and then the red nucleus will tell you whether the cells are in the G1 or S phase, and then the cytoplasm, the change in the color will tell you that caspases are activated. default it is not opening as a. This is actually that's what I showed, that a growing tumor expressing the cell cycle marker and cell death marker. If you very closely you see that the whole, there is it's almost 200-300 micrometer size. You see in the middle actually there is a core sheet. There is no cells actually. It's not be created without cells. You cannot. It's actually the tumors as and when grows, it start dying from this. And so that basically surprised. We are surprised. We thought that it's died by apoptosis actually, but you will find there is no fluorescence because the cells are dying by necrosis. So this debate that the just like clinic actually necrosis is most common in the growing tumors. So we are just recapitulating that. Then again I will show. Anyway you are going to have a very wonderful talk on various technique of super resolution, and what you see here is that what is this? Mitochondria actually. Okay, the mitochondria most often in live it is mitochondria you have seen only with a transmission ultramicroscope. Picture looks like almost same, and this is the two proteins were expressed which goes to the mitochondria, and we image in live cells using a super resolution microscopy, structured illumination microscopy. So that is what we are reaching. The resolution is this is a structural illumination which will only have a 100 nanometer resolution, but still is taking the resolution of the mitochondrial crystal structure very well. But there are techniques that will be explained to you later. With this I will stop. Okay, essentially what I was trying to tell you that there are two separate things actually. A lot of revolution is happening in the microscopy sector, from wide field to fluorescence, then confocal microscope. Then now we are going to an area of super resolution technology, which is still is just growing and growing and growing. And a separate microscopy technique is also emerging called light sheet microscopy. That will tomorrow you will find a different kind of microscope than what you see nowadays. It will look different actually, obviously. So that kind of changes are happening in the microscopy in the last 5-6 years actually. Similar to that, a lot of developments in the other aspect of fluorescent proteins and dyes development, and linking these nowadays any kind of questions which a biologist wanted to know can be addressed very well and precisely. That's all. Questions? Very important question. Very important questions. Each and every experiments when you do it, you need to know the behavior of cells very well with respect to the imaging conditions you are providing. I know this. The way in which we used to manage is that the cells are excited with a particular power 100 times. This will die. If you know that, you try to limit below 100. And now certain times you cannot do this. The best thing is that here how you optimize it. You have a large number of options. If an image which you have seen is taken with 100 millisecond, each image 100 millisecond, you can reduce to 10 millisecond. The way how you can do this is that you use a detector which is more powerful than that. And then excitation light you reduce significantly less. Then you can increase it. So every time when you do an experiment, you must know how much the cells will tolerate with the excitation you are giving. And there are ways to reduce that. If there is too much damage, there are ways. You use another camera. Then again, when you use a camera also, you can compromise on certain things. Calcium imaging, what I have shown here is that 600 sequences over a period of 12 minutes. If you wanted to get a good image, here, a visually good image, you need to expose 200 or 300 milliseconds. But why you want a good image here? What you are looking? You are looking the change in the ratio, the quantification. So don't worry about the quality of the image. You just worry about the information you are going to collect. Reduce the exposure time minimum as possible. Then again, the resolution is there. You don't have to bother about the resolution. The resolution, if you are not bothered, then what? The camera will allow you a lot of options. There are camera which comes with the EMCCD options. You increase the gain of the EMCCD so that you can significantly reduce exposure time, and you can bin the camera. Instead of 1020 frame, 1000 to 1000, you can reduce 500 and 500. You can reduce the pixel. Those options are there actually, and that's what I saw a very interesting talk here actually. Optimizing, isn't it? There is a talk, optimize imaging conditions. Something was I saw, and there you will find large number of options to manipulate and manage. First is you need to know the biology of the cells, how much it tolerate, and then the potential of the system you are using. Then you can have a play. Obviously, if a bad camera is used, that in order to at least to get an image, it has to be exposed to 1 second or 2 seconds. 3 images is fine, it will die. 3 seconds, if you are imaging 3 seconds with around 5% of the mercury lamp, 5 images cell will die. But with that time, you can, if you have a good camera, 5000 sequences also can be collected. If you have a good camera, that's what camera will cost from 2 lakhs to 1 crore. Here they say, I don't know about
They're expensive. I know that.
So those things are these are very important. This knowledge is very important. When you go with more and more advanced application and imaging, you need to know more and more about those things as well. It's a very important question. Okay. Yeah, then anyway, I just will show you. I will explain you that with the green fluorescent. None of the dyes which are currently used for ROS detections are good, not good. None of the dyes are good, because these dyes also specifically work on ROS generation. They automatically generate ROS. It is not because of the treatment actually. Now, basically, Roger Chen himself developed, knowing this, developed a Redox Sensitive Gfp. I just will show an image of that. Yes. This is actually a Redox Sensitive Gfp. It's actually having a unique property of is an actually excitation specific dye. We have 405 and 488 excitation. Emission is always in the green color. When it is oxidized, it will show more emission at 405. When it is reduced, it will show more excitation at 488. See, this is actually a ratio which is shown over here. After a drug treatment, this is actually a basal level of ROS. After treatment with the drug, when it is oxidized, see this color change, and more, it's actually a time lapse of oxidation. More and more color. It's actually a live cell compatible fluorescent green fluorescent protein based sensor of ROS. This also one of the beauties that this can be targeted to any organelle. You wanted to do your mitochondrial ROS, you want to cytoplasmic ROS, you wanted to nuclear ROS, you can image like this. See the heterogeneity very precisely. In some mitochondria is highly oxidized, some mitochondria is not oxidized. The small changes in subcellular changes also can be visualized by this technique. This is one of the wonderful probe, but you need to express inside the cells. Dyes are, we have used almost all the dyes which are currently available. None of them are good actually. Good, provided you will do very well knowing that weakness of those dyes.
Any fluorescent dye producing
Fluorescent dyes. Dyes means? No. This is what I, no. Basically, there are people who are getting dyes from other companies and then conjugating, and atto dyes are now, some point of time there was a Bangalore based company called, they were not really generating the dyes. They are getting the dyes from a specific company and they are conjugating actually. We don't have a company which specializes in generating dyes or proteins. Huh?
To start a spin-off in India.
Yeah, definitely, it's a huge potential, but no, problem is that we are nowhere actually, So if you begin with you are somewhere now, is fine, but we are nowhere. We are not started anywhere, anything. So this is, Russia is doing very well in green fluorescent proteins. Japan is doing very well. Yeah, see, slow dividing, and this is difficult, slow dividing, and if you try slow dividing, and the way in which you call this actually, is relative actually, 3 days slow dividing and fast dividing cells, you can identify this, but there are techniques called, there are certain dyes actually very powerful. Slow dividing cells can be identified in a population very precisely. These are lipophilic dyes which will bind, and called PKH is from sigma, and also CFC from invitrogen. They will go and bind to the cells uniformly. As and when they divide, they will equally distribute. So as and when the generation happens is equally divided. A cell which is slow dividing will retain the dye as such. If it is not divided, the same dye will retain as such at the end of 20-25 days. So this is actually, it is called slow tracking dyes actually. These are very powerful tools. So 30 days you can track even in animals. Is something like a pulse chase, but it is called not pulse chase. There is a typical term. It's not CFC, PKH. There is a specific term. I don't remember exactly.
And anyways, like stem cell proliferation and differentiation, we can identify through.
Yeah, there are approaches actually. There are anything can be done nowadays actually. If you know that there is a marker which will tell you differentiation. For example, albumin is actually a differentiation marker for liver cells actually. Okay, if you have an albumin expression, either which is tagged with fluorescent proteins actually. As and when it basically cancers are undifferentiated. When you induce them to differentiate, that can be as a marker. Similar to that, any cells, any plant cells, anywhere you have a differentiation marker. If you express with the green fluorescent protein with the half-life must be a little bit slower, because GFP's half-life is a little bit more actually. Destabilized GFP or destabilized DS-RED, this will allow you to track immediately, differentiation immediately. You can see it.
Like say stem cell homaging.
Stem cell homing, people have used actually. People have used this actually. There are a lot of differentiation markers. Like for example, a very typical example, OCT4 has been very widely and very popular to OCT4 GFP mouse. GFP mouse is there actually, because it's an undifferentiation marker. When it differentiated, it will lose that GFP. OCT4 is lost actually. So there is a mouse. It's developed as early as probably 2000.
Is that Fred Caspase? Is that commercially available? Like your Fred Caspase system?
Okay.
Is that a commercially available product?
Yeah, this is not commercially available actually.
I didn't think so.
It's not commercially available. This is Adgene has actually one vector. It is commercially available from Adgene, but basically it's not for commercial purpose. Because recently I have tried to get a license for commercial application. No, they said no.
Really?
Yeah, it's no, because I don't know.
I mean, I can see that. Wow. I mean, I see so much Caspase stuff.
I actually
That
one
would
of
be really awesome.
exactly one of the companies asked us, but then I gone back and wrote to Mayuda. He said that this one of the fluorescent protein was developed for Thermo Fisher. So the patent belongs to Thermo Fisher. So, you cannot work on that actually. Commercial application is there, it has to come from them, but they are not interested such. So, there are a lot of for academic purpose there is no issue at all. When it commercialization, people have all concerns, even if there is a potential for it, is a real problem. Evrogen is actually they are giving actually, but they are also giving the limited license to their spin-off companies, and not like I don't know, I am not very sure about that, whether as such they have given a commercial license to anybody. Yeah, definitely. Yeah, there also you should remember. I forgot to add that actually, because real time, when we are talking about real time, then because of that only I talked about the green fluorescent protein based technique. In really any protein can be looked for interactions. Only thing you have to use 2 fluorescent dyes. One dye for one protein which acts as a donor, another dye for another protein which acts as an acceptor. You have to look for the emission from the first one has to have a significant cross excitation of the second, that is all. The Alexa 488 and Alexa 564 has been widely used. Alexa 440 and Alexa 488 has been used. Now you large number of combinations are COI3 and COI5 has been used. FITC and DS also, FITC and TRITC also has been used. Lot combinations are commercially available now.
Dr. Babita? Manikandan?
That we can adjust the resolution. So if we use single channel also, we can directly capture this image, or for SSI we use something is different. Long you have shown that pixel something. So you can select what range you need. For this is a 10x objective, so we can zoom it also and can adjust the resolution. This single channel output, you can take single channel output, otherwise pixel 1, you can use it to channel pixel 1, 3 channel pixel 1, you will like this. That will be so we can take it individually or we can and then format whatever we want, and we just capture this image because when we so this machine comes with this light with this option. Data file അത് റെഡി ചെയ്യുന്ന മുമ്പായിട്ട് ചെയ്യാൻ പറ്റും. കാരണം ഇപ്പം എക്സാമ്പിൾ വെജിൻ ഡേറ്റാബേസ് എക്സാം കുറേ സോഫ്റ്റ്വെയർസ് ഉണ്ട്. അപ്പോൾ അതിനകത്തൊക്കെ സാധാരണ റോ ഡേറ്റ ഇപ്പോൾ ഫോർ എക്സാമ്പിൾ സെൻറ്റിൻ്റെയോ എഫ്എമ്മിൻ്റെയോ റോ ഡേറ്റ ഡയറക്ട്ലി അതിനകത്ത് ലോഡ് ചെയ്യാൻ പറ്റുന്നുണ്ട്. ക്ലൈമറ്റ് ചേമ്പറൊക്കെ സെറ്റ് ചെയ്യാൻ പറ്റും. ടെമ്പറേച്ചർ സെറ്റ് ചെയ്തിട്ടുള്ള ചെയ്യാൻ പറ്റും. വേറെ ആ അറ്റാച്ച്മെൻ്റ് എല്ലാം ഇതിനകത്ത് ചെയ്യാം. കാരണം ഇതൊരു DMA8 എന്ന് പറഞ്ഞ ജനറലൈസ് മൈക്രോസ്കോപ്പ് അത് നമുക്ക് ഇതിനകത്ത് ഇങ്ങനെ 100 വരെയുള്ളൂ. ഒരെണ്ണം ബ്ലാങ്കാണ്. ഒബ്ജക്റ്റീവ് അഡോൺ ചെയ്യണം. അഡോൺ ചെയ്താൽ അത് നമുക്ക് മാറ്റുന്നുണ്ടെങ്കിൽ മാറ്റി സെപ്പറേറ്റ് സ്റ്റേജ് ഒക്കെ വയ്ക്കുന്ന പരിപാടിയായിരിക്കും. കാണിക്കില്ല ഇത് ചെയ്ത് ഫസ്റ്റ് ഓർഡർ ഫ്ലൂറസെൻസ് എടുത്തു, ട്രീറ്റ് ചെയ്തു, ഫ്ലൂറസെൻസ് പോയെങ്കിൽ പിന്നെ ഡൈ ആഡ് ചെയ്തു. SSC ഒരു സെൻട്രലൈസ്ഡ് ഫെസിലിറ്റിയാണ്. അപ്പം നമുക്ക് ഇവിടെ ഒരു സെൻ നോർമൽ സെൻ ക്ലാസ്സസ് നോർമൽ അതെല്ലാം മാറും. കൂടി നോക്കിയിട്ട് പ്രോസസ് നോക്കിയിട്ട് പിന്നെ ഈ സെറ്റോട്ട് പോകും. കാരണം കിട്ടുന്നുണ്ടോ എന്ന് അറിയാൻ കാരണം നമുക്ക് താഴോട്ട് പോകണം. ആ സ്റ്റാക്ക് ചെയ്തതിനനുസരിച്ച് സാധാരണ
Does a nice job of getting the threshold over the top.
64.
So